Am I audible? Hello.
Yes, sir.
Good evening, one and all. Yeah, today topic is nitrogen metabolism. Actually, last class we completed about oxidative phosphorylation and ETS cycle, and also balancing sheet of ATP formation. And before that, we have learned about glycolysis and the Krebs cycle, and another important photosynthesis. I have sent the video. So now the another important topic is nitrogen metabolism. So in nitrogen metabolism, what is nitrogen metabolism? Hello? Nitrogen metabolism and nitrogen fixation. Where is nitrogen first? Hello? Nitrogen availability
In the atmosphere, sir.
in the atmosphere. Okay. How it is enter into the plant? How it is entered? So anyway, here the what we learnt it during nitrogen metabolism. So as you said, the this atmospheric nitrogen enter into the plant and inside the plant associated with plant body. That is called metabolism. So the nitrogen metabolism, it is refers to the biosynthesis of amino acids in plants and animal body. That is called metabolism. So here the biosynthesis of amino acids means amino acids formation. How the amino acid formation with help of nitrogen? How we get the how the plants get the ammonia? The ammonia get from the atmospheric nitrogen. I am asking another question here. Can the plant receive direct atmospheric nitrogen? Can
No,
the
sir.
plant? No, did not receive directly. Here the atmospheric nitrogen, after convert into nitrate or nitrite, then only they absorb. That form only absorb the plants. So in this process, first one, let's see first assimilation of nitrate, and next one assimilation of ammonia. First, let's see assimilation of nitrate. Let's share the PPT. some of the missed the PPT. So anyway, the first, let's see assimilation of nitrate. Right. So actually, plants absorb nitrogen from the soil. But I already told you where are the nitrogen in general. Was the nitrogen, atmospheric nitrogen? Actually, it is not in soil, atmosphere. Actually, plants absorb this nitrogen from the soil mainly after it is this atmospheric nitrogen turns into NO3 minus NO3 minus nitrate or NO2 minus or by ammonium also by ammonium. In this form only plants root absorb, right? In this first two, nitrate and nitrite, this is by physical method observed. These are the formation by physical method and ammonia formation by biological method. So anyway, these are observed by the plants, plant root. So the plants have to reduce the NO3 minus to NH4 form for enable them to synthesize various nitrogen molecules involved in various biochemical process. So anyway, in biological process, directly ammonia form, or if the any physical process, first nitrate form, this also after reduced into NH4. So finally, inside the plant, this form, NH4 form, ammonia form present inside the plants. And this ammonium involved in formation of various amino acids or protein. This is utilized for formation of various amino acids and proteins. This nitrate reduction takes place in root as well as leaves also. So, already told you, actually the soil, inside the soil, nitrate or nitrite, but inside the plant body, ammonia. Inside the plant body, ammonia. Right? Is it clear? Next one, after formation, here to reduction of ammonium, first what happened inside the plant body? First the soil, for example, inside the soil, nitrate present is reduced as nitrite. To electron and again reduced as NH4. In first to formation nitrite, here the nitrate reductase enzyme involved by nitrate reductase. Nitrate, because here the nitrate reduction takes place, right? After nitrite reduction, converted to ammonia, here involved the enzyme is nitrite reductase. First enzyme is nitrate reductase, second enzyme is nitrite reductase. So anyway, here the ammonium formed in the plant body. After ammonia formed, ammonia assimilation. This ammonium assimilation in plant body. Assimilation means here the resulting of ammonia is then rapidly assimilated into the various nitrogen compound. What are the nitrogen compounds in plants? What are the nitrogen compounds in plants? Mainly the subunits of proteins such as amino acids and direct proteins and also nucleic acid. If we take amino acids are NH2, these are called amino acids. If you take protein, already told you the proteins are subunit of amino acid. So NH2 chain, NH2 chain, NH2, again NH2. So in this also nitrogen present. And another compound present in nucleic acid. What are the nucleic acid? Hello? What are the nucleic acids? The nucleic acids are DNA and RNA. If you take the DNA basic structure first, it is made up of nitrogen bases. Inside the DNA, nitrogen bases are there. What are the nitrogen bases? Purines or pyrimidines such as adenine, guanine, thymine, cytosine. These are the nitrogen bases. That's why in this the nitrogen compound present. And if you take the RNA also same. In RNA also nitrogen bases are there. So this is the assimilation, nothing but after formation ammonia, where these ammonia utilized for formation of plant compounds, right? So next one, nitrogen fixation. This is nitrogen metabolism. Already told you the definition. Nitrogen metabolism. What is nitrogen metabolism? Hello? Biosynthesis of amino acids. Biosynthesis of amino acids in plant. We are talking about plant, that's why biosynthesis of amino acids. How the amino acids are biosynthesized? Amino acids are biosynthesized from nitrite, ammonia. Later, this ammonia assimilated inside the plant body in the form of amino acids. It is the metabolism. Now let's see the first of all how it is fixed. Fixation, nitrogen fixation, I already told you. Physical nitrogen fixation, biological nitrogen fixation. Another one is industrial nitrogen fixation also there. The industrial nitrogen fixation through the Haber-Bosch method. Haber-Bosch method, we can get the nitrogen. Uh, in this, first of all, in Haber-Bosch method, using the high pressure 1000 bars and temperature 1000 bars and temperature 450 degrees centigrade temperature industrially, we can prepare the ammonia for by utilizing nitrogen. So this is the industrial process. And also physical nitrogen. In physical nitrogen, actually, uh, just wait, can I rough it? In in physical fixation, what happened? Of the 2000 million metric tons of mud, that means most of the nitrogen uh, fixed annually by this natural process only. Another one is physical, nothing but the natural process. The natural process mainly occurs by the raining and thundering and lightning. What happened during this period? First, gas nitrogen, first of all, gas nitrogen, gas nitrogen combined with oxygen, in atmosphere, combined with oxygen, form nitric oxide, form nitric oxide. This nitric oxide, these are formed in physical method in environment. Nitric oxide further oxidized, as well nitrogen dioxide formed as nitrogen dioxide. This is physical method, remember. sorry, nitrogen dioxide. And now this nitrogen dioxide combined with water. Already told you this is happening during rainy lightning and thundering process. Due to raining process, waters are formed. Now after this nitrogen dioxide combined with water during the rain and produce the nitric acid HNO3, nitric acid HNO3 produce HNO3. Now the trace of the nitric acid are also produced in the stratosphere. The stratosphere is the layer of the atmosphere by in the air the by photochemical reaction between ozone and the nitrogen dioxide. The acid airs brought down the earth along with the rain, and here these after reach the soil of the seed reach the land. What happened here? HNO3. Just 1 minute. Okay. This HNO3 after mixed with soil particles, that is H plus NO3 minus, right? This NO3 minus reduced to nitrite in plants or in soil. This nitrogen in plants actually to common. Okay, this is the physical method. And the take page number 111. Do you have books? Hello?
Yes, sir.
Right. Turn on page number 111. Here, after physical nitrogen fixation, here the given the biological nitrogen fixation. In biological nitrogen fixation, some of the important nitrogen fixing organism given just by these organisms. Actually, these are the mostly useful for comparative but not theory. But anyway, the biological nitrogen fixation, if if asked in the examination, you just write about biological fixation process. I will explain now. So anyway, first in triple one, are you in triple one page?
Yes, sir.
Right. So here given the some of the important nitrogen fixation organism. First, heading asymbiotic bacteria. What are the asymbiotic bacteria? In asymbiotic also aerobic bacteria, microaerobic and facultative bacteria. Here the aerobic bacteria given Azotobacter and Dixia, Azospirillum, Azospirillum for microaerobic, and facultative in facultative asymbiotic. Facultative
What is,
asymbiotic example.
what is facultative?
I will explain this way. Facultative means here the two type. One is first one is facultative, and another one is always something named. Just wait. Sorry, obligative. Facultative and obligative. Facultative means facultative asymbiotic nitrogen bacteria. Here actually it is act as a symbiotic process. Symbiotic means what is symbiotic? What is symbiotic?
Symbiotic.
Facultative. Facultative asymbiotic. Here, facultative, here given facultative asymbiotic bacteria, right? Actually, these bacteria can fix the nitrogen fixation in general by symbiotic process. Symbiotic. Symbiotic means association with another one. That means, for example, if you take rhizobium. Hello? Do you know rhizobium bacteria?
Yes, sir.
Rhizobium bacteria fixation nitrogen in symbiotic process. Symbiotic means here in this both nitrogen, sorry, rhizobium bacterium take host from the plant body, and plant body get benefit from rhizobium bacteria in the form of nitrogen fixation, in the form of nitrogen availability. So this is symbiotic process. Here the facultative example Klebsiella. Klebsiella bacteria also same nitrogen fixation like a rhizobium. Hello? Hello? Are you follow me?
Yes, sir. Yes, sir.
Right. Here the Klebsiella also fixation nitrogen like a rhizobium. But if there is no association, if there is no association in the situation also, this Klebsiella can fix the nitrogen, but not rhizobium. That is the difference between facultative and applicative. And here the facultative simple. One thing, for example, in clear way, facultative, uh, one of the facultative for example, facultative parasite. One organism facultative parasite. First of all, what is parasite? What is parasite? Hello?
Parasites, they live in the host.
Right. Live in the host. Right.
Yes.
Here the facultative parasite means actually it is not a parasite. It is not a parasite. Actually, these are saprophytic organism. But when the host is available, then they can perform as a parasites. Hello?
Yes, sir.
Now the word clear or not? Facultative word.
It's act like symbiotic.
Yes. Facultative, here the heading asymbiotic bacteria. Asymbiotic bacteria means there is no symbiotic nature. If you take Azotobacter, no symbiotic nature. If you take Azosphaera, no symbiotic nature. But Klebsiella, actually in general, Klebsiella can fixation nitrogen with the symbiotic nature. Symbiotic nature means with another host, with help of another host, the presence of another host. But if the host is not present, if the host is not available, in that situation also, it can fix the nitrogen. That is called facultative. Is it clear?
Yes, sir.
Right. Now anaerobic. In anaerobic, non-photosynthetic and non-photosynthetic Clostridium, photosynthetic Rhodosphaerium, chemoautotrophs, Thiobacillus, and cyanobacteria, blue-green algae. Here the are blue-green algae are unicellular Glycopsa, filamentous heterotrichous, Anabina nostoc, filamentous non-heterocystus, oscillatoria. Now symbiotic. Now here the given symbiotic legume symbiosis, Phaseolus vulgaris and Pisum, that means all rhizobium species, whatever it may be, rhizobium species. And here non-legume symbiosis also given, non-symbiosis Casuarina, in Casuarina plant Frankia, Pers, Azolla anabina, Cycas coralide roots, Anabina nostoc, lichens, fungi nostoc. So these are non-legume symbiosis. Here, these are all actually, if you take in symbiotic nature, in symbiotic nitrogen fixation, can nitrogen fix, for example, if you take here the Casuarina frankia, Casuarina frankia. Did you did you see the Casuarina frankia in your in your book 112 page number?
Yes, sir.
Right, Casuarina frankia. Frankia is the bacteria, Casuarina is the plant. If you separate the both, that means if you Frankia separated from the Casuarina, the in individually in free situation, Frankia cannot fix the atmospheric nitrogen fixation in plant. That is the situation called as obligative.
Okay.
But just before you asking about the facultative, in facultative, even even the any partner, host or any partner not available in that situation also, it can it can fix the atmospheric nitrogen. Is it clear?
Yes, sir. That
Facultative
is
word clear?
facultative.
Right,
Yes, sir.
right. And also here the given the somewhat elaborated about cyanobacteria. So already here the cyanobacteria, whatever it given, you write just about that. Unicellular bacteria, filamentous heterotrichus, and filamentous non-hetero, sorry, filamentous heterocystus, filamentous non-heterocystus. These example enough. Okay. And now the
Okay.
symbiotic nitrogen fixation, symbiotic nitrogen fixation. In examination, this is the very, very important nitrogen fixation method. Important. Okay. So anyway,
Yes.
just remember these names. That is symbiotic bacteria, anaerobic bacteria, photosynthetic bacteria. They are also important. But in examination point of view, most of the part asking about the about symbiotic nitrogen fixation method and nitrogen fixation mechanism and also nitrogen cycle. They are the important. Let's see the fixation method, rhizobium fixation method. Just wait. Visible? Hello?
Yes, sir.
Radial fixations, root nodule formation. Right. So symbiotic nitrogen fixation. Here the taken example legume rhizobia symbiosis. Here the legume rhizobia symbiosis is important from both ecological and agriculture point of view. In the initial process of infection of the roots and formation of nodules, the first, the rhizobia or brachyrhizodia clusters are around the root hairs. They are the habitat of this bacteria here. Uh, Here, this bacteria, rhizobium bacteria
Sir,
habitat.
can you zoom that image?
Just wait. Now is it clear? Hello?
Yes, sir.
Just 1 minute. Right. So here these are the rhizobium bacteria. The habitat of the rhizobium bacteria, they are inside the soil, in the soil around the root hairs. This is recognition roots by particular rhizobial strains is attributed to flavonoids and lectins. Actually, these bacteria why only exactly surrounding the Lignaceae family plant? Because actually these bacteria are attracted toward the Lignase plants. What happened during this? The first from the plant body, some of the plant roots, flavonoids, 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 flavonoids and lectins, flavonoids and lectins. These two produced from the host plant body. From flavonoids and lectins, the rhizobium bacteria are attracted. The lectins may promote the aggregation of rhizobium uh, at the infection zone here. This is the infection zone. And also stimulate mitotic activity in the nodule primordium. In response to this bacteria, root hair first, this root hair curl in their tip like this. Root hair curl in, this is the second step, like a hook, curl like a hook. What happened first? Actually, the ensyl rhizobium bacteria attracted toward the Leguminaceae plants by flavonoids and lectins. Before, actually, initially, first the flavonoids and lectins are released out. For these chemicals attract the bacteria when immediately for responding, the root air shows curling like a hook. Shows curling like a hook. The root hairs curl at the tip, at their tips. Now the third step, what happens? The bacteria then invade the root hairs and the plant produce an infection thread. What happens? This group of bacteria we can see in this step enter into the root hairs and form as a thread. This thread called as a infection thread. This infection thread along with plasma membrane of the root hair. This infection thread from the root hair, epidermis, cortical cells, reach the cortical cells. The bacteria, this thread, after formation, this infection thread, the bacteria proliferate and pass through the infection thread to the inner cortical cells. On these cells, turns gives to nodule formation. I will tell you how the nodule formed. This type of nodules formed inside the cortex. So anyway, the rhizobium signals molecules that plays in key role in the induction of nodules. Anyway, after formation infection thread like this, in infection threads have the bacteria. Some of the bacteria through the infection thread reach the cortex. After reach the cortex, after settled in cortex region, what happens? In the nod factors immediately activate. These nod factors have the major role to induction of the nodules in the roots, right? So this nod factor, so for example, here the bacteria. So in rhizobium bacteria, nod factors, NOD nod factors are there. Nod factors are there. These nod factors are lipoketo oligosaccharides. In these nod factor, the genes involved in the synthesis of nod factors are called genes, not genes. Actually, these nod factors synthesized by not gene, not factors synthesized by not genes. So anyway, there are several genes associated with the nodulation process. The most important are NOD, A gene, NOD B gene, C gene, D, E, F, L, all genes. And another important gene, NOD D, has the ability to bind the specific flavonoid secreted by the roots of the plant, of the host plant. So this association becomes the transcriptional activator of the other NOD genes. So finally, which encode the enzymes involved in the synthesis of NOD factor. So what happened here? Actually, before formation NOD gene, NOD gene, NOD gene, by transcription, RNA form, translation protein form. This protein called as NOD factor. So like this, in this formation of NOD factors, not only one gene, there are given different names, NOD, different type of genes, NOD A2, NOD A, B, C, D, and FEL genes. These genes are involved for NOD factors. So anyway, after the NOD factor, after NOD factors formed, the NOD factors induce the root A deformation and cell division in cortical cells, which subsequently resulted in the nodule formation. So, with help of this NOD factor, proliferation takes place inside the cortical cells. That means rapid increase of cells. Due to the rapid increase of cells, the nodules are formed. Nodules are formed. So the nodules is a highly organized structure with a vascular connection to the root. The cells in the nodules are polyploidy. These nodules are what is polyploidy? Polyploidy means the chromosome number more than two. Chromosome number status, for example, diploid, two, triploid, three, tetraploid, four. So polyploid more than four. So this condition present in the nodules. So during the nodule development, the bacteria are transformed into bacterioids. So anyway, like this here, the nodule formation, for example, this is the one of the nodule formation here. Here also under the nodule. What happened during nodule formation? Take this nodule. Let's change color. Here bacteria in this nodule cell, bacteria, B for bacteria. Here bacteria, here also bacteria. So bacteria are present in nodule and these are transformed into the bacteria. These bacteria during the nodule development, bacteria transformed into bacteria, which are several times larger than rhizobium. Of course, it's also rhizobium, but these bacteriaids larger than rhizobium bacteria. And this bacteriaid covered by layers. Bacteriaid covered by layers. That's why this is the larger when compared with rhizobium. That means before formation nodule, rhizobium small. After formation nodule, the rhizobium large in size. So this large, big size structure called as bacteriaid. Bacteriaid. Okay. So now and are the transformation bacteria to bacteriaid is closely related to the synthesis of leg hemoglobin. Now the final step, leg hemoglobin form. A leg hemoglobin and nitrogenase enzyme and other molecule required for the nitrogen fixation. So first of all, after formation, this bacteriaid, synthesis of leg hemoglobin and synthesis of nitrogenase enzyme and other molecules. These other molecules that mean what are required for nitrogen fixation. And so, right. The next. So in bacteriaids, what are I told you? Leg hemoglobin formation, nitrogen enzyme formation, the leg hemoglobin, sorry. In leg hemoglobin, heme part, leg hemoglobin, the heme is synthesized by bacteriaid, synthesized by bacteriaid. And another meioty, that is the globin, heme by globin protein, the globin protein by host. H for host. Host means plants, leguminous plants. So this both together, bacteriaid and host both together form leg hemoglobin, right? So leg hemoglobin, this is the required, this is must and should for the nitrogen fixation. So anyway, the leg hemoglobin form. What is the function of leg hemoglobin? The leg hemoglobin control the oxygen levels. It acts as an oxygen scavenger. So and no need. Okay, right. So it is leg hemoglobin act as oxygen scavenger. Then only that means the nitrogenase enzyme. And also apart from the leg hemoglobin, nitrogenase enzyme also form nitrogenase nitrogenase enzyme also form. Actually, this nitrogenase enzyme active when the absence of oxygen. If you take the normal cell, normal root cell or stem cell, leaf cell, in cell oxygen present or absent in normal cell. In normal cell, oxygen present. If the oxygen present, this nitrogenase enzyme shows inactivate nature. Inactivate does not perform any duty to fixation nitrogen, fixation atmospheric nitrogen. Here the nitrogenase enzyme compulsory to activate of this nitrogenase enzyme. Here the hemoglobin protein important. So leg hemoglobin in heme part by bacteriaid, globin part in from host. After formation, the nitrogenase enzyme synthesized in the plant body. The presence of hemoglobin, now oxygen bind with heme. Oxygen bind with heme. That's why the oxygen concentration decrease in cell. Then immediately nitrogenase enzyme activate. Now the performance and nitrogen fixation. Is it clear? Hello?
Yes, sir.
Right. Then another one is nitrogen fixation mechanism. In the here the formula given atmospheric nitrogen 8 electrons, 8 proton, 16 ATP and form into ammonia. Let's see. Just wait. Philippines. Nitrogenase enzyme. Turn on page number 116. The nitrogenase enzyme, nitrogen fixation mechanism. Nitrogenase enzyme consists of two different protein such as such as the large molybdenum-ferrous protein contains and two molybdenum atoms and 30 ferrous atoms and 30 acid-labeled sulfur atoms. They are present in this. And smaller only Fe protein consist. Smaller Fe protein consist. So just wait. Let's share the PPT. Is it visible to you?
Yes, sir.
Right. Right. Change color. Okay. Right. So this is nitrogenase enzyme. A nitrogen enzyme consists two different part. One is large. Remember, for example, this nitrogenase, large one, and another one, small. Two parts. In large, molybdenum, ferrous, and smaller only Fe protein. So this is nitrogen. Okay, right? So in this nitrogenase, some of the active sites are there. These are the active sites. These are the active sites. These active sites bind by to nitrogen. Change color. N N. That means N2. Atmospheric N2. Atmospheric N2. NN bind by. Right? And at a time here the two protons and receive two electrons. This is reduction. Reduction. And again, two protons, two electrons. Here also two protons, electrons. Here also two protons. So how many total protons? Two, 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 two. And every time two electrons also receive. While receiving this electron, it is reduced state. Always nitrogen is reduced, and actually this is formed by reduction and oxidation. When the electron received a reduction process, and immediately the electrons leave, lost the electron and oxidation. So the process of reduction and oxidation, the finally formation 2 moles of ammonia. Finally formation 2 moles of ammonia. For formation of 2 moles of ammonia, here the requirement, how many protons here? How many protons here? Here two, 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 two. Total number of
6,
protons. Listen here. 1, 2, 3, 4. 4 into 2.
8,
8 protons. How many electrons?
8 electrons.
Right. Nitrogen plus 8 electrons plus, and here for receiving two, for each reaction 2 electron 2 protons. Apart from that, 4 ATP, 4 ATP also utilized. 4 ATP also utilized. Here 4 ATP, here also 4 ATP, here also 4 ATP, here also 4 ATP. Now tell me the total number of ATPs utilized to formation of ammonia. How many? How many ATPs? Sorry, not 4 here. Right, right. 4 ATP. First reaction, second reaction, 4 ATPs, 8. Third reaction, 8, 12. And last reaction, 4. Total, hello,
16.
16 ATPs. As a result, formation into 2 moles ammonium or ammonia form. So this is the nitrogen fixation. So nitrogen fixation means how the atmospheric nitrogen convert into ammonia, but which mechanism? So with help of nitrogenase enzyme. So first of all, we see how the nitrogenase enzyme formed during the nodule formation, hemoglobin formation during nodule formation. After formation nitrogenase, then only the nitrogens fixed inside the plant body. So that is the mechanism of nitrogen fixation. Is it clear? Hello?
Yes, sir.
Hello? Right. So
Yes, sir.
nitrogen fixation also completed. And tomorrow, I think
Nitrogen
tomorrow
cycle is last class, I think.
only one class remain or two class. How many class remain?
No, don't know, sir. I think one only.
I think one was, so next class with ecology. Okay,
Okay, sir.
ecology ecosystem. I will complete in one class, but before that, you selected the topics of the ecology because the only one day there. Okay,
Okay,
you
sir.
selected topic. The topic only I will explain.
Okay,
Okay,
sir.
please inform tomorrow in
Okay.
class in lab.
Okay, sir.
Okay, thank you all.
Thank you, sir.